Hello, my name is Flay Abdulrazak and I have a background in computer science and PhD in biomedical um and I'm working in a neuroinformatics lab, and today my talk is about uh, the causal effects of cingulate surface area on brain executive functions. Executive functions are the collection of cognitive processes responsible for behavioral cognition control, including attention, inhibition, and working memory, etc. Up to this date, we do not have the neural basis of the structures that cause the individual differences in the executive functions. One of the most relevant structure that is associated with the executive functions is cingulate cortex. For this study, we are using the structural correlates of the cingulate cortex, and as we are using the data from Human Connectome project, and the output is in free surfer format, and the free surfer divides the cingulate cortex in these four subregions. Two are the in from anterior cingulate, then you have posterior cingulate, and then you have isthmus. And also we are using three tasks to measure the executive functions. We have card sort, that is to measure the cognitive flexibility, flanker to measure cognitive inhibition, and inhibition control, and list sorting to measure the working memory. Also, we have data of for the subject between age 22 to 36, and 47.5% of these subjects are male. And also we are using some other variables like handedness and gray matter volume um, to do the confounding control, and we are including them in the analysis as confounders. Mm, so we had 1,113 subjects, and we excluded because this data include a pair of mono and dizygotic twins. So to avoid any kind of biasness, we excluded one twin from each pair of twins, and the remaining 830 subjects were used for this analysis. After pre-processing and cleaning, we have established a causal DAG to test our hypothesis for the causal relationship between the surface area of cingulate cortex and the test scores. And here is the causal DAG. We want to check the average treatment effect, and these are the confounding factors: gender, age, race, handedness, family, to test to check for any environmental and genetic common factors, and also total gray matter volume. And we used the marginal structure models and inverse probability of treatment weight. What it does, that IPTW creates a pseudo population with no confounding, and this is achieved by assigning some weights to each sample, and then using this pseudo population to fit the outcome model. And our findings were that even though the initial association was significant between all the and test scores and cingulate, the margin structure models, only two models were significant: alpha 0.01, which were posterior cingulate cortex with flankers and list sort. And what we found out that unit increase in posterior surface area will result in an increase of 0.004 scores in the task for the flanker and list sort. Here you can see that. And also we have implemented e-value to test or account for sensitivity analysis. And also you can see that the highest e-values are for these same test scores, which have a significant p-value. And these were our key findings, that the singular surface area actually causally, when we account for all the confoundings, the only association or possible causal relationship is only between the surface area and flankers, and posterior surface area and flankers and list sort. and that will be all. Thank you.